Hello and welcome back to our second installment of Survey of Biology for Respiration. And in the first segment we looked at respiration and kind of the big picture, how it relates to photosynthesis, the products it creates, how organisms rely on respiration to produce the energy they need to carry out metabolism and basically live. And now what we want to do now is we want to look at a specific type of respiration that takes place in the absence of oxygen, or an anaerobic respiration. And the first process actually takes place whether oxygen is present or not. So it's kind of the start of both aerobic and anaerobic respiration. So we're going to look at glycolysis. So it is a universal process. No oxygen is required, so it is anaerobic. And if we look at this flow chart, it shows kind of in the simplest terms what's happening. So we have an input of glucose. So from photosynthesis, from the food that we eat, glucose goes in, and what we produce are two molecules of NADH, which if you remember from the first segment, we called an electron carrier. And it's going to carry the electrons and the energy associated with them to the electron transport chain, which we'll see in the next segment when we talk about aerobic respiration. We also get two molecules of pyruvate, or sometimes you'll hear it referred to as pyruvic acid. This is where the carbon ends up at the end of this process. So the carbon from glucose stays in the pyruvate. And then finally, we get two molecules of ATP. This is what the cell can actually use for work. And so whether it's chemical, mechanical, or transport work, this is what the cell really wants to get. And under anaerobic conditions, if oxygen is not present, this is the maximum. We get two molecules of ATP, and that's it. Now, if you are a very simple yeast cell or bacteria, that's probably enough to carry out what you need to live. But if you're a more complex cell that needs to move, and maybe a nerve cell, a muscle cell, you're going to need a lot more, and anaerobic respiration may not be enough. But for right now, this is what we're going to concentrate on. And so the products are two molecules of NADH, two molecules of pyruvate, and two molecules of ATP. So here is the process in a little bit more detail than what we really need to know. It's actually a very complex 10-step process. And as you can see here, it goes through, there's enzymes that will catalyze each of the steps. But what I want you to look at is there's basically 3 steps. Okay? And step number 1 is the energy investment phase. So we've got to add 2 molecules of ATP to get things started. So it's kind of like investing money so that you can earn more later on. So that's the first step. It's the energy investment. The second step is the splitting of glucose. So here's where we take a 6-carbon molecule of glucose and we split it into 2 3-carbon molecules. So we haven't lost any carbon yet, but we've split it into 2. And then the final step, here's the payback. Here's where we get ATP and NADH for the cell to use at later steps. So even though it's this complex 10-step process, what I want you to concentrate on, what are the products? And the products at the end of it all are going to be 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate to go through. That's what we need to kind of focus on, and that's kind of the big picture. So again, here is kind of the simple one. So we have the energy investment phase. So we're going to put in a little bit of energy before we get started. We've got the splitting phase. So here's where the original 6-carbon glucose gets broken in half. And then finally we've got the energy payoff phase. Now one thing that I want you to look at is here it's saying that there's 4 ATP produced in the energy payoff phase. Well, how many did I tell you last slide do we actually get? Well, if you remember, we only get 2, so why is there 4 shown here? Well, there are 4 that are produced, but if you remember back up here on the left, we had to invest 2 before we started. So what this means is that we get a net. This is what was actually there for us to use at the end. of 2 ATP. So make sure you understand that there are 4 ATPs produced, but we've got to pay back the 2 that we used during the energy investment. And then again at the bottom, I'll just kind of emphasize, we still have the 6 carbons that we started with. So 2 times 3 carbons in the pyruvate gives us the 6 carbons of the original glucose. We haven't lost any carbons yet. And this diagram kind of shows the entire process again. So we're talking about glycolysis. It's taking place in the cytoplasm. So we haven't even entered the mitochondria yet. And it is anaerobic. No oxygen is involved. So again, we've got the energy investment phase. We've got the splitting phase, which isn't really shown. And then finally, we've got the energy payoff phase. And again, down here, you'll see net. And that's what I really want you to, so we have 2 ATP that we invest. We have 4 ATP that we get back. So we have a net of 2 ATP at the end. We also have the 2 molecules of pyruvate and the 2 molecules of NADH as we move along. So that's the first part, glycolysis, and that's universal. That's going to happen no matter if there's oxygen present or not. Now we get to the step where a decision has to be made. So now we've got our pyruvate molecules from the end of glycolysis, and now if oxygen is not present, so if we have those anaerobic conditions, we're going to go to a process called fermentation. Now everything over here on the right we're going to save for a later discussion, and right now we're going to look at what happens if there's no oxygen present. And it kind of depends on whether or not we are in animals or if we have something like yeast that'll give us ethanol or ethyl alcohol. And so this is where we're going now. We're going to look at what happens when fermentation takes place. And you may recognize some of these products of fermentation. So fermentation is the anaerobic breakdown of glucose. So this first part up here happens regardless, and we end up with pyruvic acid. If there's no oxygen present, then we go to fermentation. And most of these organisms here are bacteria. So lactobacillus, you may recognize from yogurt. E. coli, Escherichia, right there, you may recognize. And all of these products, from cheese to vinegar, wine, and beer, are products of fermentation and the anaerobic breakdown of glucose. So one thing that I want to stress is that these products, these processes, after pyruvic acid, there is no additional ATP produced, which is a common misconception. Some people say, well, we'll get additional energy. But fermentation doesn't give us any more ATP. What it does is it recycles NAD+ so that glycolysis can continue. So the process, or the main goal of fermentation, is we want to recycle NAD+ so that we can go back and do more glycolysis. And I think the next slide will show that one a little bit better. So here's what happens in the case of maybe yeast. So here's what's called alcoholic fermentation. So glucose to pyruvate right up here. Here's glycolysis. And we get our ATP that we're interested in. Now we have our pyruvate. Well, if there is no O2, what happens is it gets converted, we give off CO2, and it gets converted to ethanol or ethyl alcohol. Now look what happens. If we follow the arrows, there is no new ATP. But what does get produced during this process? What gets produced is NAD+. And if we have NAD+, glycolysis can continue, which will produce more ATP from that process, and fermentation can continue as well. So fermentation gives us no new ATP, but it does recycle NAD+ so that the process can continue. So when wine or beer is brewed, this is the process that's taking place, alcohol fermentation. Now, what happens in humans or animals for this process takes place if there's no oxygen? So if you've ever been running or lifted, you may get that burn in your muscles from the buildup of lactic acid. So lactic acid fermentation allows muscles to produce ATP without O2. So if there's no oxygen present, the same thing happens. And it's a short-term fix. It leads to something called oxygen debt. We can't sustain it for very long. So again, if we look, we've got glucose going to pyruvate. So here's glycolysis. It gives us those 2 ATP, but if no oxygen is present, the pyruvate that's left over will be converted to lactate or lactic acid. And again, there's no new ATP. All that gets recycled is NAD+. This allows glycolysis to continue. This allows us to produce some more ATP through that process, but the actual lactic acid fermentation doesn't produce any new ATP, but it allows us to continue glycolysis. And again, it's kind of short term because eventually, as you know, we need to breathe. And our last slide here as we wrap things up is just kind of flow chart that shows everything. So again, the first part we talked about, glycolysis, is universal. It happens in all cells from bacteria to giraffes to whales, redwood trees and everything in between. The bottom part is where we really get to the anaerobic. If oxygen is not present, we're either going to carry out lactic acid fermentation or ethyl alcohol fermentation depending upon the organism. And that's kind of the wrap up for anaerobic respiration. Please join us for next time as we look at aerobic respiration and what happens when oxygen is present.